Hello students. Today we'll be studying the third week of development. So after this lecture, you should be able to describe the formation and fate of the primitive streak, which is necessary for the formation of the 3 germ layers. Then you should be able to describe the formation and fate of notochord. You should be able to describe the process of neurulation, describe the development of somites and intra-embryonic coelom. You should be able to explain the embryological basis of congenital malformations, nucleus pulposus, sacrococcygeal teratomas, and the neural tube defects. And you should be able to describe the diagnosis of pregnancy in first trimester and role of teratogens and alpha fetoproteins. So for our ease, I've divided it in 3 major parts. So firstly, we'll be studying the formation of the 3 germ layers. So firstly, what are the major events of the third week of gestation? So the third week of the development, it begins after 5 weeks of the onset of the last menstrual period. That is, if we count it from the day of fertilization, then you have to add 2 more weeks to the last menstrual period. So that is why it is known as third week. And if we are counting from the last menstrual period, so it corresponds to the 5th week. So that is why it begins after 5 weeks of the onset of the last menstrual period. Then the most characteristic event occurring during the third week of gestation is gastrulation. That is the process that establishes all 3 germ layers, that is ectoderm, mesoderm, and endoderm in the embryo. Now, the germ layers are the primitive building blocks for the formation of organ rudiments. So that is why this stage is very important. So the characteristics of the third week of development is firstly the formation of primitive streak, then the gastrulation, that is the formation of the 3 germ layers, ectoderm, endoderm, and mesoderm, then the formation of notochord, formation of allantois, the formation of neural plate, neural tube, and the neural crest cells, formation of somites, formation of intra-embryonic coelom, and the development of the chorionic villi. So we'll be studying this topic under our 8 headings only. So this is the end of the second week. If you remember, we had, that was the week of 2s. We had the formation of the 2 germ layers, that is the epiblast and the hypoblast, 2 cavities, the amniotic cavity and the definitive yolk sac or the secondary yolk sac. Then there was formation, the trophoblast had divided into the 2 layers, that is the outer syncytial trophoblast and the inner cytotrophoblast. Then there was also formation of the extra embryonic coelom, which divided the extra embryonic mesoderm into the somatopleuric and the splanchnopluric mesoderm. So this was the major events which were occurring into the by the end of the second week of intrauterine life. So we have bilaminar germ disc. Now what happens during the third week? You get a short note on gastrulation. So what is gastrulation? The process by which a bilaminar embryonic disc changes into a trilaminar disc during the third week is known as gastrulation. Now initially it was a plate, but later on as the epiblast will spread, you imagine a sphere, a ball. So the plate, it changes into a disc. So now we have a trilaminar disc. Now how the gastrulation will occur? So this starts with the formation of a primitive streak. So after about 15 days of development, a thickening containing a midline groove forms along the mid-sagittal plane of the embryonic disc. Okay, we are seeing this embryo from top. So this one is the amniotic cavity and we are viewing the embryo from the top, from the embryonic pole. So over the course of the next day, this thickening is called the primitive streak, and it elongates to occupy about half the length of the embryonic disc, and the groove is called the primitive groove, and it becomes more deeper and more defined. Now, the cranial end of the primitive streak is expanded into a structure which is called the primitive node. It contains a depression in the center which is known as primitive pit. And from this, the cells are continuously expanding caudally. So the formation of primitive streak, why it is important? Because it defines all the major body axes of an embryo. So it divides the embryo into a cephalocaudal axis. Over here, this one is the primitive streak. Okay, this one is the longitudinal section of an embryo when we are viewing it from above, and this one is the cut section. Now the primitive streak, it forms in the caudal midline of the embryonic disc. Caudal, that is, it is occupying the posterior half of the embryonic disc. So it will define the craniocaudal axis as well as the medial and lateral axis. Okay, so when we are viewing from above, the um, and also left and right axis. So if we view it from above, the left will fall on our left side and the right side of the embryo will fall on our right side. Now the primitive streak, it regresses by 26th day of the intrauterine life. Now, what happens after the formation of primitive streak? The epiblast cells, they will start migrating towards the primitive streak. So the migrating cells, they will start invaginating into the primitive streak and they will start detaching from each other. So the movement of cells through the primitive streak and into the interior of the embryo is called ingression. So ingression is also known as invagination. So over here in this diagram, this one is the primitive streak, this one is the primitive node, and all the epiblast cells, they are entering through this and entering inside the embryo to form the definitive endoderm. So cell migration and specification, they are controlled by fibroblast growth factor 8, which is synthesized by the streak cell. So that is why primitive streak is called as the main organizer of the embryo. So over here, this you can see this one is the primitive node. The epiblast cells, they are my detaching from each other. They are changing their shape. They first become flask shape and they start migrating into the interior of the embryo and they will start displacing this hypoblast cell layer to form the definitive endoderm. So the first layer which was formed was ectoderm. Then the first layer to be formed within the embryo over here or after the ectoderm, what is formed is the definitive endoderm. So what is the cellular basis of the primitive streak formation? So there is a theory which is known as Collar's sickle or a robber's sickle theory. What happens? There is a thickening of an extra embryonic tissue at the caudal edge of the germ disc. So this one is the germ disc. The caudal edge, it will get thickened. The extra embryonic cells over here will get thickened. So this will induce, so this thickening is known as Collar's sickle. So this Collar's sickle, it will induce the adjacent epiblast cells to form primitive streak by cell-cell interactions at the caudal end of the embryonic disc. तो सबसे पहले ये पीछे से बनना start होती है. And what are the cellular bases of the primitive streak formation? It involves 4 major processes. One is the cell migration, that is the epiblast cells, they will start dividing and they will start migrating towards the center. And then there is oriented cell division so that it will form a linear streak in the mid-sagittal plane of the embryo. There is progressive delamination from the epiblast. So until unless they will delaminate, they will not be able to migrate. So progressive delamination from the epiblast. And then there is convergent extension because it has to extend mid-sagittally and craniocaudally only. and it occupies only the half of the embryo. But it is said that primitive streak, it remains of same length, only there is a growth of the embryonic disc, and all the cells which are dividing, they are migrating towards the center, which will leads to its extension. So that is why the convergent extension of the primitive streaks of the four major processes which forms the cellular basis of the primitive streak is the cell migration, oriented cell division, progressive delamination from epiblast cells, and then there is a convergent extension of this streak. Now the formation of the three germ layers. Now the invaginated cells, they will start displacing the hypoblast cell layer to form the definitive endoderm. Now after the next layer, after the endoderm, the next layer to be formed is the intraembryonic mesoderm. And the remaining cells of the epiblast, which will not migrate inside, they will form the definitive ectoderm. Thus, epiblast, it will form all the three germ layers by gastrulation. So that is why it is known as mother of all the three germ layers. So migrating mesoderm, it separates ectoderm from endoderm except two places. Cranially, where the ectoderm and endoderm remains adherent, is at the buccopharyngeal membrane, which corresponds to a precaudal plate. If you remember, some of the hypoblast cells, they became columnar in the second week. So it led to the formation of precaudal plate. So that corresponds on the embryonic disc to an oropharyngeal membrane where the mesoderm will not be formed. So oropharyngeal membrane cranially and cloacal membrane caudally are the two places where the ectoderm and endoderm are closely adherent and mesoderm is not formed. So what is a buccopharyngeal membrane or the oral membrane? It is a small oval depressed area at the cranial end of the germ disc, and it appears in the precaudal plate. In buccopharyngeal membrane, endodermal cells are firmly adherent with the ectodermal cells. It is bilaminar and it is devoid of mesodermal cells. And it breaks in the fourth week to form the opening of the oral cavity. Now, what is the cloacal membrane? At the caudal end of the primitive streak, the germ disc remains bilaminar due to firm endodermal attachment with the ectoderm. Later on, further development, cloacal membrane divides into anal and urogenital membranes. So the cloacal membrane disintegrates in the seventh week to form the openings of anus and the urinary and genital tracts. Then the mesoderm also differentiates into a pericardial bar. So it is a midline horseshoe shaped mesodermal cell condensation which will be cranial to the buccopharyngeal membrane. So this is a summary to tell you the process of gastrulation. So firstly, by uh, oriented cell migration, there's cell division, convergent extension, and process of delamination, there is a formation of primitive streak, which is induced by a Kohler sickle at the caudal end. This one is the caudal end because here is the cloacal membrane. Then there is a formation of the Henson's node, which is the cranial most part of the primitive streak with the blastopore, and there is a primitive groove formation. Then there is migration and invagination of the epiblast cells, or there is a process which is known as ingression, which leads to the formation of definitive endoderm, intraembryonic mesoderm, and finally the ectoderm. So all the three layers, they will be formed in the third week. So bilaminar regions in the trilaminar disc are the oropharyngeal membrane, which corresponds to the precaudal plate, and the future position of the cloacal membrane at the caudal end. Now, what is the cellular basis of the gastrulation? The process is known as epithelial to mesenchymal transformation. That is, epithelium is well organized structure. They are well adhered to each other by EK adherent, which is a receptor. Then, if it is down regulated by certain factors like fibroblast growth factors, the epiblast will start changing its shape. They will become flask shaped, they will delaminate, and they will start migrating inside. And it will form a loosely interwoven structure which is known as mesenchyme. So this is known as epithelial to mesenchyme transformation, which is leading to the formation of definitive endoderm and finally the mesoderm. So epiblast cells lateral to the primitive streak, they begin to move into the primitive streak where they undergo an epithelial to mesenchymal transformation. So it involves certain process, that is epiboly, which is known as spreading of an epithelial sheet by mitosis. Then emboli is ingression or internalized movement of the cells into the interior of the embryo. Finally, there is convergence, that is all the cells from the embryonic disc, they are moving towards the midline, that is convergence, which will lead to convergent extension. And finally, the lengthening of the craniocaudal plane. Why? Because all the cells are moving towards the midline and it cannot be accommodated into a small space. So there is lengthening into a craniocaudal planes. So certain fate map are established during the gastrulation. So the cells which are ingressing from the cranial region of the node, they becomes the precaudal plate, which has already been formed. And the school says the first cells which are ingressing through the primitive node and they are displacing the hypoblast will form the precaudal plate. And another view say that the certain hypoblast cells, they will elongate, they will become columnar in second week and they will form the precaudal plate. Both are correct, nothing has been confirmed. Then the second cells which are entering through this primitive node, the cranial end will form the pre-notochordal cells, which will form the notochord. Then those migrating at the lateral edges of the node from the cranial end of the streak will become paraaxial mesoderm. Now the paraaxial mesoderm will give rise to the somites and the head mesoderm. So this HM is head mesoderm, S is for the somites, which is given to the paraaxial. Paraaxial is meaning near the axis, adjacent to the axis. So mesoderm is now then organizing into three parts: the paraaxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. So cells migrating through the mid streak will form the intermediate mesoderm, and that migrating more caudally will form the lateral plate mesoderm. So they will organize into the disc. Along the axis will be the paraaxial, then adjacent to it will be the intermediate, and lateral most will be the lateral plate mesoderm. So, and the cells migrating through the caudal most part of the streak over here, it will contribute to the formation of extra embryonic mesoderm. Along with another source was the primitive yolk cells, that is the hypoblast cells. So some of the interesting facts about the third week is the embryonic disc. It remains bilaminar at two places, that is the buccopharyngeal membrane, which corresponds to the precaudal plate, and the cloacal membrane. There is continuous migration of cells from the primitive streak towards the cranial region, which makes the disc elongated craniocaudally. And cilia of the cells of primitive node are responsible for right-left visceral side determination. So if there is abnormality in the cilia beating, then it may lead to a condition which is known as situs inversus. That is, major visceral organs are reversed from their normal positions. Now is the formation of notochord. So what is notochord? You get a short note on a notochord. So notochord is a midline embryonic structure that develops during the third week of development from the cells of primitive node. Now, what is the extent of the notochord? It extends from cranial end of the primitive streak to the precaudal plate. So, first migrating cells, excuse me, from the cranial most part of the primitive node will form the prenotochordal cells, and they will organize from the cranial part of the primitive streak till the precaudal plate. So, over here you can have a look. This one is the primitive node. These are the prenotochordal cells. They are migrating. Over here, this will be the precaudal plate. So, it will organize itself cranial to the primitive node. So this will form the notochord. This is the full formation of the notochord. Here is the cut section. This one is the cut section. So over here, this one will be seen at the transfer section of the embryo at this place. So this is the notochord. The blue one will be the ectoderm. The red one will be the mesoderm, and the yellow one will be the endoderm. So this, the tri-laminar germ disc has been formed, and here the notochordal process has also been formed. So this corresponds to the late 17th day. Now next step will be the formation of the notochordal canal. Here, as you all remember, this was the primitive node. With the Henson's node is a depression inside it. So it will also extend into the interior of this notochordal process, and this will form a notochordal canal. So by 17th day, by the migrating epiblast cells, they will form a notochordal process, and there is an extension of the canal from the pit. The primitive pit will extend into the interior of the notochord as a notochordal canal. So here the stepwise formation of the notochord is given. Firstly, what happens? The epiblast cells, they will migrate inside. They will form the notochord process anterior to the primitive node and caudal to the precaudal plate. They will form a notochordal process first. Then it will arrange into a notochordal plate. It will then detach from the underlying, it will first forming a notochordal plate. Then its lower cells, they will uh, they will fuse with the underlying endoderm. So now it will give rise to a notochordal plate. Now the flat notochordal plate is being formed. Now a transient neuroenteric canal will be formed, which will provide nutrition to the developing ectodermal cells. Then again, there will be a folding of this, the embryo will start folding also, and laterally. So this will lead to the detachment of this notochordal plate, and it will lead to the rolling over itself, and now it will form into a solid bar, which is known as notochordal process. So the transformation of notochordal process includes around 7 days, which extend from 16th to 22nd day of the intrauterine life post fertilization. So firstly, what happens now, I'll discuss the events in detail. That is, the cells of the primitive node knot, they will proliferate. Over here you can see the proliferation of the primitive knot cells. Now around day 17th and 18th, the proliferated cells invaginate in between the ectoderm up to the precaudal plate to form a solid cord cells, which is known as notochordal process or the head process. Now what happens in the second diagram, you can see the lower layer of this notochordal cells, it is now disintegrating at some places, which is forming the neuroenteric canal. Now I already told you there is a cavity which is known as primitive pit. It will extend in the interior of the notochordal canal along with its elongation. So this is known as the notochordal canal. That is, the extension of the primitive pit inside the notochordal process will, as a canal, is known as notochordal canal. Now the cells of the notochordal canal, that is the lower part of the cells, they will fuse with the definitive endoderm. Now they will convert this notochordal canal into a neuroenteric enteric canal. Okay, so cells of the notochordal canal, they will disappear in the craniocaudal direction to form a communication between the amniotic cavity via primitive pit with the yolk sac. This communication of amniotic cavity with the primitive yolk sac is known as neuroenteric canal. Now, the neuroenteric canal flattens to form a notochordal plate in the roof of the yolk sac. So, it incorporates firstly with the endoderm. What happens next? Soon the flattening of the notochordal plate reverses by the folding of notochordal plate. It will start folding upon itself, and the cells of the notochordal plate get separated from the underlying endoderm to form a solid cord of cells called the definitive notochord. So, firstly, there is migration of the cells to form a notochordal process. There is extension of the primitive knot as neuroenteric canal. Then there is fusion of the craniocaudal fusion of the notochordal cells with the underlying endoderm to form a neuroenteric canal. Then after that, this will form a notochordal plate. Then this notochordal plate, it incorporates with the underlying endoderm. Later on, it will roll upon itself to form a solid notochord. So what is the significance of the notochord? Notochord is a characteristic feature of phylum Chordata animals. It defines the axis of the embryo and forms the basis of the developing axial skeleton, specifically for the vertebral body. In humans, notochord disappears except its remnant. In adult, it represents nucleus pulposus of vertebral discs and apical ligament of dens, which you will read in the head and neck. So two remnants of the notochord are nucleus pulposus and the apical ligament of dens. So this is the summary of the formation of notochord. That is firstly the proliferation of the cells of primitive knot, formation of a primitive pit, then the formation of neuroenteric canal, formation of notochordal process, then finally formation of notochordal plate. Over here, this is the formation of notochordal plate, then rolling over itself, then it is detaching from the underlying endoderm and it is forming a definitive notochord.